Good evening, everyone. Thank you so much for joining this very special CMU alumni spotlight series with Kaustubh Deshpande, who graduated from CMU in 2018 and has a very, very interesting journey from graduating CMU to joining Elon Musk's Neuralink and now starting his own company, Eywa Neuro, that manufactures neuroelectrodes and brain machine interfaces, brain chips. I think the way the world is going, the way machine learning and AI is moving fast, what Elon says that, we need to be cyborgs and we need chips in our brain to keep up with AI. I think to see it's a very proud moment to see a CMU alum who's working on hardware. Not, we see a lot of software startups, and hardware is the hardest. So it's great to see Costa, working on hardware, working in India, making in India, and selling to the world. So Costa, welcome, and thank you everyone for joining this from our CMU network. This session is going to be interactive. So as Costa goes through his slides and presents his work, we will do a interactive Q&A session with him as well, where he can delve deeper. So the goal of this is for it to be interactive, so you can unmute yourself and ask your questions, or turn on your videos whenever you want to, since it's a very close group event. So Kaustubh, welcome. It's a pleasure to have you. Glad you joined the Mumbai group. And please take a few minutes to introduce yourself and tell us more about your journey from CMU to Neuralink to Eywa before we jump in.
Sounds good. Thanks, Ashay, for your kind introduction. You've been pretty kind in the way you talked about me. Sometimes, and you forget about what you did in the past. You're so focused on the future that it feels good to look back and have friends and fellow colleagues appreciate the work that's been put in. But and thank you everyone else who's joined the call. I know it's the end of the day for a lot of you, and despite that, you've taken some time out to listen to me, and let's try to make this presentation fun. We'll have a good interaction. Pontificate about the pros and cons of having chips put into our brains, and we'll see where it goes from there. So first things first, I came across a funny meme just recently where healthcare has always been big business for donkey's years, right? And then initially, people started with organs that were more elementary to understand. So, gallstones and kidney stones. And now the focus has been on brain machine interfaces and neurosurgery. A lot of the money has been pumped into it, and dopamine's been making a lot of money lately. So, if the younger ones are trying to find, a niche in biomedicine, I would probably definitely urge you to try out neurotechnology, neuroengineering. So my journey was, I never planned to be in this field. It just happened, call it luck or call it destiny. But I as an electrical engineer, I joined Carnegie Mellon to focus on devices, semiconductors. But somewhere down the line, when I was at Hammerschlag Hall, the famous building on campus, I chanced upon an opportunity to do research with Dr. Nesam Chamanza, who was a new professor there back then, and he was starting his lab at CMU. He had just moved from UC Berkeley, and his focus was to make neural electrodes, so implantable neural electrodes that can record electrical activity in the brain, and perhaps stay in the brain for a long period of time. So my work, I was able to publish a couple of papers while I was there, and my work got recognized and allowed me to get a job with a company called Neuronexus. So if I back up into history a little bit, Neuronexus was the first company born out of University of Michigan at Ann Arbor that commercialized invasive neural interfaces. So they'd been doing work since the late 80s, and they were the first commercially successful company that made neural interfaces. So it was a great opportunity for me to go in and work with them. They're a small company, so getting opportunities was it was tough, and I got lucky, be it timing or the work that I'd put in at CMU. But I worked at Neuronexus for 3 years and did a lot of research work on making chronic neural interfaces, chips that could be put into the brain that would stay in the brain for long periods of time. And that work, and at one of the conferences, it got me an opportunity to interview at Neuralink. One of the co-founders there, Dr. Vanessa Tolosa, she knew me and she was familiar with my work. So I interviewed there. I actually got a chance to interview with Elon because we were a pretty small company back then, and now it's grown to a large size. But I was on one of the initial teams when they just started scaling their operations. So at Neuralink, did a lot of groundwork to develop the interfaces that they had now. It was right from it because it was a small team. It was all hands on deck basically, and it was some of the work that we did was right from tooling and buying tools, taking care of tools, to building processes, doing iterations on design. It was a lot of work, heavy work, but it was a lot of fun. And one of the glory moments that happened for me there was the media presentations in Gertrude the pig and Pesha the monkey. Those
Yeah.
implants were made by me. So
Oh,
I'm
wow.
always I'm always gonna carry that badge of honor wherever I go, because they've got my name written there as the engineer who fabricated them. But that because of the work that I put in there, and the passion that I had there, I was selected to be one of two engineers to work on the next generation of the implant. So we did a couple of trials and errors with that, but eventually focus was still on the first generation and taking it through the FDA route. So unfortunately, COVID struck in 2020, and you know times were pretty uncertain. So it due to personal reasons, family reasons, I decided to move back to India and continue working from here. And those were tumultuous times, and nobody knew what was going to happen. But fortunately, I got a lot of support from the Indian government as well as the Center for Nanofabrication at IIT Bombay. They allowed me to come in, mess with their machines, develop processes, and soon enough, by the end of 2021, I had a robust process flow where I could make my electrodes and then sell them. So for the first 6 months, I continued doing that, and I still worked for a few companies in the US. I did consulting work for them, and I did contract work for them. And eventually, I realized that I don't have to be tied down. I got the confidence to start my own company. And I said, let me try and sell this to everybody across the world, not for a select few companies. So I did that, and in the next 3 years that followed, I was able to export these devices to Israel, to the US, to the UK. My company started getting recognized for the custom work that we did, and basically we created a niche for ourselves because it's such a small area of expertise that somebody does it in India, it's likely that it's going to grab attention because the economics of it
Right.
work out, let alone the tech. So the work that I did got recognized by the Ministry of Electronics and IT in India, and my company was awarded by them for a contribution to the Indian semiconductor mission. So as I stand now in 2025, the goal is to continue working on what I started with, that is to build a neural interface for humans. And so far, we've got two patented ideas, and we're now on the road to doing clinical translation for one of our devices. Now, I know these are all complicated words, so I'm gonna slowly get there and try to introduce to you what a neural interface is, how it records, what it does. It's gonna be pretty interesting. So before,
Awesome.
hindsight is always 2020, right? And as a younger person 4 years ago, I didn't think of this when I started it. Probably for the best, because ignorance is bliss. But I came across two nice quotes, and I'm going to start with them. So this is by Naval Ravikant, and he said, "Best problems to solve are the ones that you can't stop thinking about," and kind of my trajectory followed the same path. I continued to want to push on the neural interface route because nobody has done it before, and it's equal opportunity to everybody. And the second thing is, if I realized on route to starting a business, that when you have a leverage, you can really utilize that and build your business, build capital, uh, get people to work for you, build technology, and it starts compounding. So these two thought stayed with me, and that's kind of what I'm still building upon. So now going
That's
to the
also
tech
by
part.
Naval, right?
Yeah, that's also
That
another
that's
Ravikant. Yeah,
yeah. Okay,
yeah.
cool.
So, all right, I'm going to get into the science of things now. What you're seeing right now is a mouse brain. Okay, it's what you see. It's really a tangle, of wires. And to understand what a brain does, you need to understand what each and every wire does, right? So you can imagine that a small animal like a mouse has such a complicated brain. Can you imagine deciphering the human brain? It must be tricky. We've got 84 billion neurons. Neurons are the smallest cells in the human brain. And to record and identify each of them is a monumental challenge. So much like the Human Genome Project that started in the 90s, where researchers tried mapping different gene sequences, a similar project called the Human Connectome Project started in the Obama era, where there's a lot of money pumped in to really reverse engineer what the brain does. Because much like an electrical appliance, the brain is also electrical in nature. So the idea came in that if a computer can think in terms of electrical signals and our human brain can think in terms of electrical signals, then why not make an android? Why not have the two communicate with each other to make communication, processing, everything faster? But before we get to that stage, we need to understand which parts of the brain does what. And that's a complicated challenge. And that's where things began in the late 80s. So to give you an understanding of why this research is critical, uh, neuro-related problems are the second highest disease burden, in the world. There are over 35 million people just in India living with these problems, and all of them can be mitigated to some extent with the help of these invasive neural interfaces. I think a lot of them have been coming to the forefront now with people speaking out and social media being out there. But earlier, people didn't speak out about them. They would just suffer in silence. So really, to solving this problem would really help people who are in need of them. And some of the diseases that can be solved are epilepsy, Parkinson's disease, depression, urinary incontinence. And I'm not making light of the statement that I'm about to say, but I've actually seen people undergo treatment with invasive interfaces, and their tremors that they used to get with Parkinson's disease completely stopped when they had the interventional therapies. So
Really?
really, it's yeah. So it's not a pie in the sky idea. This has really been done. It's been FDA approved. And it's just a matter of time that this becomes more of an accessible treatment, as opposed to something that is still niche and people don't know about. I think it's going to take effort from all corners of this problem, right? Surgery.
And just also a quick question on that. You said it's FDA approved
Yeah,
via
yeah.
Neuralink or via the work that you're doing. Like, what's the
No, no.
which companies are doing this?
So Medtronic does this. Boston Scientific does this. This is a therapy
Okay,
called deep brain stimulation. So they make electrodes for. They've been making. It's very similar to the pacemaker technology. So
okay.
it took inspiration from the pacemaker technology in the 50s, and then they started implanting something similar in the brain, towards the 90s. But it's only gotten widespread approval very recently, like in the 2010s, maybe. So it's still as not as well known as it should be, but it's gaining. The number of people seeking this treatment are growing exponentially, not linearly. So, the brain, unlike just a regular electrical appliance, doesn't have wires. They've got axons and dendrites, and there's some element of biochemistry happening as well. Unlike wires or just the electrical phenomena that we see, where current is conducted by electrons, in the brain, it's not electrons, it's ions that are doing the charge conduction. So, when you say you're low on electrolytes, those electrolytes are what are actually helping you think. Those are the nerve pulses that pass through. So you've got sodium, potassium, chlorine that enable you to process and send signals. So when the brain thinks about something, let's say I want to lift my hand, the brain does the firing of the synapses, and then the spinal cord receives them, and the spinal cord acts as a bus. It then distributes it to the rest of the body, where the rest of the body starts picking up. Okay, I need to move my hand, or I need to move my thumb. So every instruction is broadly mapped. So if I compare my brain with Ashay's brain, it's going to look small in comparison. No, I'm just kidding. It's going to look very similar, physiologically, but ideally, when it's dynamic in nature because evolution happened that you can't just have one pathway for one instruction. But broadly, most instructions that the human body understands happen in the same areas between people. And so now, eventually, in the 50s, there was a technique called lobotomy, where people would insert, an electrode up somebody's nose and just kind of scramble around in the brain, and that was dangerous. So people realized that to move forward in this field, you need to get better tools. You know that popular saying, right? That an engineer is as good as the tools he uses. So it's true in this case as well. So people said, all right, let me take electrodes and let me put them on my scalp and on my head. That technique was called EEG. And they realized that the signals that they recorded from them were from a lot of parts of the brain, and they were jumbled. So it was very difficult to decipher them. If I wanted to pinpoint it to a certain region, it was quite tricky. And that's because if you look at the anatomy of the brain, you've got the dura, which is very thick skin matter, and then you've got bone above it. So any electrical activity basically gets suppressed. It's such weak electrical activity that it's hard to make sense of it. So people said, you know what, let's get closer to the source. So they took the bone out and then they said, let me put it on the top of my brain. And that technology was called ECOGs. It's still used today in a lot of neuromonitoring applications. But again, they face problems with getting very good spatial resolution because they were still quite far off from the brain. Finally, somebody said, let's make something so thin that it can pierce the brain without damaging it or without making it bleed, you know, bursting through all the blood vessels in the brain. And we'll start recording activity from different neurons in the brain. And that technology is called invasive, intracortical neural probe technology. And you can see that the signals on the right start getting clearer and clearer. So, what you read here doesn't make a lot of sense. The ECOG slightly get better. And then as you go further invasive, you can see independent spikes, and that gives you the best resolution possible. So to give you a comparison of the different modalities that are out there on the right, you can see that benchtop MEAs or penetrating microelectrodes give you a resolution of up to 100 microns. So you can resolve neurons that are 100 microns in, spaced apart by 100 microns, as compared to EEG, which is over a centimeter. So it's, different applications require different resolutions, but when you're doing surgery or when you're doing PCI, you really need extremely high spatial resolution. Otherwise, you're going to misinterpret everything and nothing makes sense. And at the same time, the tools for recording these signals became important to the backend tools. But people realized that, okay, the regular amplifiers that we used cannot be utilized for recording such small voltages. So they started focusing on making analog front end that records very minute signals that can amplify them, digitize them, and then we read the digitized data so that it can be stored, it can be processed, and it can be recreated. So, in an ideal case, what the engineers would call a closed loop feedback is when the electrodes record something, the electronics processes it, and then we give instruction back to the brain. That's the ideal case, right? It's like a telephone where both people are speaking. Now, this is a great part of history, and I wish more people knew about this. But and I'm going to give you like a naive example for it. I'm sure a lot of you, when you were younger, just closed your eyes and said, "All right, I'm going to use all the force I have, and I'm going to think, think louder or think in a more stronger way." And it doesn't make sense, right? Like you can't do much. You can't tangibly, understand or sense much when you do that. But in reality, you can modulate your brain, and this discovery was made in 1979 by a professor called Fetz. So he was working with monkeys and he gave monkeys rewards every time the monkey did an instruction. And he realized that eventually he started upping the threshold. So if the monkey thought with a certain frequency, the number of pulses per second that the monkey is shooting, he would reward the monkey. And he realized that the monkey can modulate the rate at which he thinks, the rate at which he fires synapses, which was great because now for somebody who has a well-functioning brain but has maybe challenges with his spine or limbic system, you can pick those signals up and you can train a computer model to understand them. So this is where the ML part comes in. This is where a decoder reads these signals, decodes them based on the action, and then moves an external stimulus, be it a cursor or be it a keypad, some external stimulus that so that that external actuator mimics what the brain is thinking. And this is exactly what we did at Neuralink. When we inserted this in Peja the monkey, he was playing a video game, or what he thought was, what he thought like he was controlling with a joystick. But in reality, it was the chip that was recording this, the decoder reverse engineering his thoughts, and then the computer cursor playing them out on the screen. And there was no lag. There was no, the monkey didn't feel any different as opposed to when he was just playing with the joystick. So this really was the founding principle of brain machine interfaces, that a person can modulate his firing rate, and based on that you can leverage that fact and create very high efficiency brain machine interfaces. Any questions so far?
That's a good point. I think this was a lot of good information. Anyone has any questions, you can please unmute yourself. We are in a closed group for CMU network as well, so you can just ask the questions when you have them. Anyone
I
with
have a quick
questions?
question. Yes,
Sure,
hi.
Weber.
So, just to understand this one a bit deeper, so are we saying that if we provide some external stimuli, which is,
Mhm.
let's say, more dense, which I
Right.
understood what you were saying from, thinking deeper.
Mhm.
So we can basically then sort of change the actions of the monkey? Or
No,
did I
well,
completely butcher it?
no. So basically, the point was that you can modulate your thought. So when you think, right,
Mm,
there's a string of pulses called synapses that get fired. And
right.
ordinarily, like let's say I want to move my hand just up and down, there would be maybe, let's say, 10 pulses that get fired. But I
Mm,
have the control to increase that rate of pulses. So I could maybe try and hit 15 pulses or 20 pulses, or I could reduce them down to 5 pulses. So that concept that you
mm,
can modulate the activity of your brain really, the field took off with that finding that, all right, if I tell a human that, okay,
mm.
think, but I want you to think with this firing rate, you could identify exactly where he's doing so, or the part of the brain that
I
he's doing
see.
that from, and you could modulate that into action. So in the monkey's case at Neuralink, we had implanted the chip in his motor cortex, which is the part of the
Mm,
brain responsible for moving his hands and legs. And
mm.
we took that data from we recorded data when he was playing with the joystick, and we realized, okay, these are his firing rates, this is what he wants to do with the cursor, and we decoded that with machine learning, and we could reproduce that, by giving instructions to the computer. So that's how you could pick up just raw electrical activity from the brain and translate that directly into a secondary actuator's actions.
Oh, that okay. Now this is very clear. Thanks for
Yeah,
that. And just follow up on that. So let's say if a monkey is, doing something with the hands, and
right,
another scenario, the monkey is doing something with the legs, walking or something, right? So
right.
basically, I'm assuming that different parts of the brain would be sort of, working there, or is it the same thing but different electrical signal frequency? Like, how does it
That's
work?
a that's a good question, and it changes. So, back a little his insight into history. As humans evolved, the way that the brain grew wasn't linear. So you don't find
Mm,
the same sort of cells or the same sort of signals in all parts of the brain. Initially, the brain was only meant to control our digestion. Can you believe that? Because the
mhm,
digestive part of our body was the most complicated for in the human body. So the coordination between the colon, the intestine, the colon, the stomach was the most complicated activity it did. But as humans evolved, more and more actions started coming in. So we got a temporal lobe which coordinated time, any basically time coordination, or we got a
Right.
motor cortex which is responsible for our limbs. And there are a bunch of things. So parts which are responsible for eyes, for the ears, the auditory cortex or optical areas. So all of them are different physiology. So that's why there's no one solution fits all. It's very custom, and there's no linear answer to your question.
Got it. But I did get it just. Thanks so much for that. Yeah.
No problem.
Hey,
Right,
I have
moving
a.
forward. Sure, sure.
Can I bump in with a small question? So, does that
Certainly.
mean that if you have a neural link implanted in your head,
Yeah.
you can still have private thoughts?
I mean, yeah, because this is just electrical activity, right? And unless the decoder figures out what you're thinking, you're safe. There's no, it's unlikely that you could
Okay.
think about your ATM pin, but unless
Huh.
you go in and put it in, it's unlikely to pick it up.
Yeah, straight into Black Mirror territory here.
Yeah,
And
it's
Kaustubh,
pretty
just a
cool. Yeah,
quick one on that. That when you say
yeah,
firing rate of thought, that just means
yeah.
concentration.
Well, no, I mean, we don't know how it works. It's biology, just like we don't know how the stomach works, right? It's just part of biology. It's some part of our brain that allows us to modulate this. We don't know how it does it. We don't understand the underlying principles, but we can record what is happening. We can record the result of it. So,
Yeah,
what
because
you're seeing on,
what you said was interesting,
yeah,
right? Like because the brain was used for the digestive system. Like the
right.
gut has its own neurological system. Like that's where the
Uh,
gut instinct comes in from, right? Like it has its own
right.
neurological system. So then I guess the body evolved for the brain to do something different and the gut to do something different then.
Yeah, I mean, this happened over thousands of years, right? So it
Yeah.
just kept adding and adding until, because if you look at evolution, the we evolved from primates, right? And if you look at the evolution in primates, when their bodies shrunk, their heads also shrunk, or when their bodies grew, their heads grew. But it's only in the case of humans that our bodies shrunk, but our heads kept growing bigger. So we're the only one, and that's why when a human baby is born, it cannot balance its own neck. You don't see
Right.
that anywhere else in nature. The size of our heads is abnormally large for the body we carry. And another interesting thing in this research came out of Israel. They identified that as evolution happened, we lost a lot of features as well, functionalities as well. Like the Neanderthal man was more violent than we are today. Had stronger arms, stronger legs, but that doesn't, so the brain still evolved, and we still have that information somewhere encoded in our brains, as part of how we evolved, but we don't use it. That's why there's that popular analogy, right, when they say we only use 20 to 30% of our brain. That is really true, because a lot of it is still you know genetically encoded with information that, or activities that our ancestors did. Now, it's a controversial theory. I cannot say this with certainty. Some people believe it. They call it the silent neuron theory. I like to believe. I'm a proponent of that.
And then do you think these chips can activate the remaining 60-70%?
Yeah, it's possible. People. So one experiment that happened in. I saw this at a conference. I cannot recall who did it, but they excited the pleasure centers in a rodent brain, and they realized that the rodent, even if the rodent couldn't control himself, like even up till like the point where he could die, he just wanted to be excited all the time. So as living humans or as living creatures, it's very abnormal that somebody could do that, right? Like they want to get that activity until they die. But it's clear that there's some parts of the brain which, when stimulated, they like being stimulated. The body has no control of them. So it's also kind of evidence that a large portion of the brain still have encoded information which we cannot directly control or see in our daily lives.
Very interesting. Go ahead.
Awesome. So now let's get into engineering, right?
Sorry to interrupt. I think we have
Oh,
a
sorry.
question for
No
Vivek. I
problem.
see your
Yeah,
hands up.
sure,
Vivek?
sure.
Yeah, yeah. So these interfaces convert the electrical signals into some sort of data points that we can
Right,
analyze and say what the person was thinking or doing. So
right.
in the end, it is mostly like spatial and temporal information, right? Which neuron fired
That's correct.
where and what was the sequence of firing. So
That's
this is
correct.
data point is converting for analysis. So what
Mhm.
AI models do you use to take this raw data to
Right.
something meaningful? Like what do we have some name, anything saying, hey, these are the type of models we use?
That's a good question, Vivek. I'm unfortunately not the right person to answer this but from my elementary understanding of how it works. So if you see on the left, right, this is the kind of map, the space-time map that we create, and each of these blips that you see are basically action potential or spikes that we call. That's where they're being recorded. So depending on the density of these, we pass them to a decoder. Now, here's the tricky part, right? Because going to my previous comment where the brain is not linear. It doesn't have the same cells or the same uh, physiology everywhere. We use certain designs on our neural interfaces that can allow us to triangulate where the activity is coming from. So depending on both the geometry of the brain and the specific design that of the neural interface that's being put in, we can then use certain models. Now, there are a few by a company called OpenEFIS. They've got some spike sorting models that they use, but I don't know too much about the machine learning aspect of it. I'm more into the electrical aspects of it.
Okay.
Yeah. All right. So
Thanks.
now I'm going to get into my territory, which is how do you engineer these interfaces? Because when you look at the brain is very concentrated. The spacing between two cells is less than 80 microns, which is, I think, a fourth of the diameter of a hair. So how do you put something in that doesn't destroy the whole thing and still is able to record from parts of the brain? So we use techniques adopted from the semiconductor technology. So how they make transistors, MOSFETs, or how they develop ICs, we use a similar technology, but we make mechanical elements out of them as well. So very, very thin films are deposited on silicon wafers, and they're designed in a way that they can record this activity. They're very small, so they're less than 10 microns in thickness. 10 microns meaning less than maybe a tenth of the diameter of a hair. So if I show you the cross section, this is what it looks like. There is some structural support. Then there are two pieces of insulation, and between them there are conductors. So on a single substrate, we've got multiple such points, each corresponding to a different point on the probe, which can pick up independent electrical activity. And now we use materials which are biocompatible. So we've got platinum, we've got gold, stainless steel 316, and very recently people have been working on polyamide. Polyamide is a high temperature flexible material that is biocompatible and very inert, so the brain doesn't destroy it. All right, so now I'm going to get into the history of neural probes. So after the pacemaker, which was a revolutionary technology, started, uh, hey, guys, just give me one moment. I think my screen is frozen.
Sure.
Right, sorry about that. So the history of neural probes began right after the pacemaker revolution started. So people used semiconductor technology to build very, very minute probes. So here you can see polyimide driven cables in 1988 that were made. There was and the University of Michigan and John Hopkins were two places where a lot of this research happened. And each of them had a different design. They would target different parts of the brain. They would have different ideologies. So some would have a spiked electrode, which would kind of go in and sink in the brain and not move. Some would have a flexible design that could, move with the brain. Because when we breathe, the brain also moves, a couple of millimeters every time we do that. So that you don't want your device to get displaced in the brain. So you want something that moves with it, which is why flexible devices come into being. This is a nerve sleeve, so you could put a nerve through one of these and record from it. So this is how the field started. Now things started getting interesting because in the 90s, a lot of research started coming in, and people started identifying different parts of the brain that corresponded to activities. So be it moving the arm or moving the leg. So in 2006, Brown University, along with the University of Utah and a company called BlackRock Neurotech, uh, together the collaboration was called BrainKit. So they implanted these devices into paralyzed humans, and then with the same topology, they were able to move a robotic arm basically to do their functions. And you can see monumental moment. This made the papers. A person who couldn't even grab a drink of water would use a robotic hand to go and get food or grab a drink for themselves. And for a normal person, this is a very trivial activity, but for someone who's in a wheelchair, this is huge. It solves a lot of their problems, and it marginally improves their quality of life. So now 2010s, 2000s, we started getting into high density neural interfaces. High density meaning that now, so far, we've been recording from one electrode or two electrodes, but now we wanted to put as many electrodes as possible on a single device. So you can see here that people started moving up, 16, 32 channels, 64 channels, 128 channels, 256. Some did 1,000. Neuralink was doing 1,024 electrodes per device. And then some even, so far, I think the best uh, density was done by a company called Paradromics, where they put 64,000 electrodes in the brain. So you can imagine, just like the Moore's law, which where ICs started getting smaller and smaller, you can pack more and more electrodes per device. And people then started experimenting with different modalities. So they would add fluidics to it. So you could do targeted drug delivery. You added optical stimulation to it for robotics. And then new companies started coming up because they started getting acceptance in the clinical field, and different technologies started coming up. Today, we've got an actual chip. So the company called NeuroPixels, this does only research. They make electrodes only for research, but you can actually put 4,000 electrodes with a chip inside the brain, and it does beautiful recordings. And it's extremely thin. This came out of IMEC in Europe. And this is the Neuralink device. So on the right is the first generation Neuralink device, where we use the USB cord to tether the implant to our recording interface. But eventually we said, you know what, we're going to make it so small that it's going to have a battery, it's going to have a Bluetooth module, and it can wirelessly transmit and receive data, with the outside world. So a person who got the Neuralink implant looks perfectly normal. There's nothing sticking out of them as opposed to when BrainGate did it 20 years ago. And the density of electrodes is also way higher. As you can see, we call them neural threads here. Each of these small golden rectangles are electrodes. And these threads are implanted with the help of a robot that goes in and places these. And you can see how thin they are. They were less than, I think, 40 microns in width. So extremely thin, don't damage the brain whatsoever. I'm going to just show you a quick video here of Pager the monkey, who who learned how to play this ping pong game, uh, with a joystick. But then the information that we recorded from his brain, we were then able to replicate it, and the monkey played the game without realizing that his joystick was tethered. I'm going to skip to this part. You can see the joystick is no longer connected. It's just the information we're recording, we're decoding, and the monkey doesn't even realize that he's playing the game. When Neuralink implanted this in a human, he was able to play Mario Kart, right? Can you imagine? Just as opposed to just moving a robot, to grab a glass of water, a person's now able to play Mario Kart on his Nintendo. So the technology jump was phenomenal.
Fascinating.
And now this technology came out in Australia, where people said, why put these devices in the brain? Implant the devices in the brain, where you could pass them through blood vessels in the brain. So here they've made something that looks like a stent. They call it the stent rod. They pass it through an artery in the neck. so that it's very, very, it's almost like non-invasive if you think about it, very minimally invasive. And it can go in and it can record data from the surface of the brain. And this company doing extremely well, by the way. So anyone's looking at investment opportunities, I would highly urge you to look at this. They were able to do some BCI studies in humans and actually get very good results. This is, I think, Australia's leading BCI. They've got presence in the US as well now.
What phase are they at?
They've actually developed this technology, so they're at a point where they're gonna commercialize this. They've finished with, I think, clinical trials as well. And they've got a few publications on it. The only challenge with them is that they need multiple modalities, because here you're making a compromise with the spatial resolution, right? Because the electrode is just sitting, on the top of your brain. You're not able to get very, very crisp readings. But with the help of, let's say, an eye tracking software or speech software, they're able to get pretty much the same result with a much less invasive surgery.
So,
Hi,
how
this is
Someone's got a question.
I think Devang has a question, right?
yes. Sorry if I interrupted anyone. Sorry. This
No
is
problem.
Devang. Hi, I'm based here in Mumbai. I'm in the venture capital field, and you talked about the investment angle. So just a bit
Right.
curious from that standpoint. Are these? It's a very high level question. The investment opportunities in the space, are they classic software or a mixture of software and hardware? Is hardware commoditized as it is in many other industries, and the true IP lies in the software, or is it somewhere in the middle? How do you characterize the attractiveness of investments from a venture capital standpoint?
That's a good question. And if you look at the market, right, it's huge. So there's the market potential is undeniable. But I think the premise is that it's a combination of the hardware and the surgery technique. I wouldn't put the software in there, because software problems have been solved, in the early 2000s. People were able to use algorithms to reverse engineer this, and they're more or less classic in nature. So they have the similar traits. But the way these interfaces are designed and the way the surgery is conducted brings the novelty between the companies. So if I show you the Neuralink, implantation, you can see that the monkey looks perfectly normal, right? The surgery is so crisp. It's done with the help of a robot, and there's nothing, the dangers from implanting these are very minimal. Because the robot is so precise that it misses even the blood vessels in the brain. It's that tiny. Whereas when you look at Synchron, it's a different opportunity, because they're making a compromise somewhere. They're making it, you don't have to cut open the brain, but you're still getting decent enough recordings that it can be used, for PCI. Now, having said that, and again, legally I can't advocate for anybody or dismiss anyone, but I think the technology delta comes from the hardware and the way the surgery is done. Because here, for Synchron's case, you still need a neurosurgeon. With Neuralink's
Okay.
case, it's done by a robot, and they're trying to make it so fast that it'll be like LASIK surgery, basically, where person goes in, 40 minutes later, he's done with the surgery, and he walks out. With Synchron, it's a different deal. There's more manual intervention involved. There's a compromise with the recording. But however, having said that, people still would prefer to get this. If you ask me personally, I would prefer the less invasive option. So, there's a little bit out there for everyone.
Okay.
So some
Great. Thank
surgeons.
you.
I
have a
think,
question.
yeah, go ahead, please.
Yeah, so all of these methods are talking about wiring the surface of the brain, but there are many regions which are far deeper. And how do you manage to interpret those signals? Are they important?
That's a very good question. Unfortunately, the reaching. So if you look at the number of people or patients who have problems in the deeper parts of the brain, they're a fraction of the total patient population. But because this is an invasive surgery, it's still very difficult to put this in deep structures, because to reach the deep structure, you have to kind of go through bulk of the brain. The brain is like, a piece of dough, right? 3 dimension. If you imagine a piece of dough, to reach deep parts of the brain, you have to really kind of cut open a lot of the brain. And there is a trade-off, you could lose functionality. Some surgeons do not prefer to do surgeries in those cases. Some, try to do that, but it's still a problem that needs to be solved in this field. A lot of these are still surface problems. So the electrodes that you saw, they're able to go maybe about 10 to 15 millimeters deep, but not beyond that. There are efforts for solving that problem, but it's still a tricky, problem to solve.
Okay, so we've kind of classified which are all things that you can do by just tapping the surface and trying to do
Yeah.
that better.
That's right.
Okay.
That's right.
I'm guessing you'll talk about, or maybe I missed it. I was walking part of the way. Which areas are the ones that is just motor control kind of?
Yeah, so motor cortex is a big focus area, because most people who need this surgery are paraplegics or quadriplegics. If you get their limb movement back, they're more than happy. Their quality of life improves significantly. The ones that want to play video games with these, it's a little far out, right? So the first patient target patients are people with who are paraplegic or quadriplegic. I think that similar technologies can be used. It's a platform technology. So with different designs and different clinical data, you could then target diseases like blindness or deafness. They can be solved to some extent. In fact, there was a company called Second Sight in the early 2000s that actually used something similar to give partial vision back to people who were blind. So this, because the brain is electrical in nature, you could really solve a lot of these problems. Also, one more point I wanted to add for Devang. Uh, I recommend these as good investment opportunities because the economics of them are quite favorable. Much like semiconductors, when you build a batch of these, because they're very tiny, the cost to build one implant is very low. And when you do the surgery, the ability to garner revenue is quite high. So this makes it very promising.
Right. So we should just take it offline. It's a very interesting
Sure.
model, but it's
Yeah.
new because I haven't seen or closely identified a company with a differentiated hardware and a surgeon intervention. So, but let's take it offline. It's a longer discussion.
Certainly, no problem.
Right. I think there's more questions, but let's move ahead, Kaustubh, and then later on we can
So-
take the additional questions.
Sounds good. All right, so I'm going to quickly go through how the processing is done. So much like the semiconductor technology, we use a CAD layout tool to draw out the design. So as a company, I make these devices for people who are doing research in different animal models. Each of them requires a different design. The idea stays similar. Much like ICs, we draw out the different layers that need to go in. We then make a photo mask because the device is built layer by layer in a clean room, in a microfabricated clean room. So this is what a typical wafer looks like. Each wafer gives us, 400, 500 to 1,000 devices per run. So every run that we do, we can make, thousands of devices, much like ICs. And then we use technology called lithography, which basically allows us to, with the right optics, it allows us to pattern very, very fine features. So with the technology I have, I can pattern features down to 4 microns. That's how thin my dimensions can get. But with what Intel is using or what TSMC is using, they can pattern features down to 5 nanometers today. There are a lot of the new age chips that come in they're so small. So you can you can just imagine where technology is going from. In 2014, we were using 180 nanometers technology. Now we're down to 5. So truly, like tremendous technology jump. Uh, Again, I'm probably going to skip through this. I just wanted to show you pictures of what these instruments look like. They're made by German and Swiss companies who are great at optical instruments. But with different printing modalities, lithography modalities, you could make features down less than 10 nanometers today. Then to get the films, right now, when I say I have metal lines that are running on these devices, you would probably ask me, how are you able to pattern such thin films, right? Such thin, films of metal. So the answer to that is we use a technique called evaporation or sputtering, where we take a small amount of metal, we evaporate it, all right? We heat it so much that it's evaporated into fumes, and it then goes and condenses down on the wafer. So with this technique, we're able to get deposition thicknesses in the range of nanometers, anywhere from 10 nanometers onwards. We get very good deposition thicknesses. And ideally, if you think about it, you don't need a very thick wire to go in, right? You need very thin films. So that's how, this is what an evaporator looks like. It's got a crucible, we heat it so that it vaporizes, then it goes and forms, the film on the wafer. And this is the silicon wafer, much like ICs. This is where we manufacture them. All of this is done in vacuum. I wanted to emphasize the why it's critical is because a speck of dust is 25 microns in diameter. So if it lands on one of my device, it's going to kill 50% of my yield. So that's why we need to do this manufacturing in clean rooms where the particle density is less than 100 particles per cubic foot of area. It's expensive to run a clean room, but in India we've got clean rooms set up by the government, by the Ministry of Electronics and IT. They're there in different in leading institutions like IIT Bombay and ISC, and some that are used by defense organization in Chandigarh. This is an image, an optical image of one of my devices. I had made this for implantation in a moth. You can see that each of these openings, right, these electrodes, the small yellow circles, they're only 15 microns in diameter. And I have to be very precise when I pattern them. So this technology, basically, plasma etching technology, lithography, and MEMS fabrication allows me to do that. So this, from where we are right now in the commercial world, in the medical device world, everything is hand assembled. We're now moving, to an order of magnitude that is almost 100 times smaller than that. So that is the technology jump and why this technology is widely appreciated too. To bond, okay, I'm probably going to skip through this. Feel free to ask me questions about this later, that more details about the manufacturing technology. Just last few slides on what my company does. When I came to India, I realized that the problem isn't about developing a new technology. All the commercial companies use the same MEMS fabrication technology to build these devices. The difference sometimes is in the materials that they use or in the designs that they have. So when I came back, I studied material chemistry to a large extent. In fact, I'd been studying that since my CMU days. And I came here and I started manufacturing my own chemicals. So it's biocompatible now. I export it. People are using it to build their commercial interfaces. There's a company in the UK that uses my polyamide material for building their neural interfaces. And then we've also started doing a lot of assembly work. So India as a nation, when you look at Apple coming into India and assembling the iPhones, this is a great area for us to grow into. So this is also something that my company is doing, assembling very small devices. That's a nice image of a wafer that I'd done. So all of these lines are basically metal that's patterned, and then each of these are different devices on the wafer. They're used for different target applications. Some are used for spinal stimulation. Some are used for deep brain recording. And the different cross sections here. My devices are so thin, they can be scaled down to a thickness of 5 microns. So you compare that to the BrainGate array, which is 300 microns in thickness. Mine is almost 1/20th of that. It's very similar to the Neuralink technology that's out there. Only difference being that I make my own materials. Just some images of, close-up images of the devices that I make and how they're patterned. So one more technology that I developed is making an adhesive film for this electrode. Because if in the regular world, when you're trying to stick two things together, it's easy, right? You put glue on one and take the two and slam them together. But when you're working with such thin films, if you want to put them on any substrate, right? In this case, it's a glass fiber. You have to come up with a way to pattern the adhesive of them. You can't put them by hand because they're so small. So I came up with a technique. It's a patented technology by my company to do this, and we're trying to get this into the commercial. The slide talks about the effects of stimulation. So the idea is that earlier on in my talk, I talked about bilateral communication being key in brain-machine interfaces. Stimulation is a technique where you force an electrical synapse to occur in the brain. So you're actually passing instructions to the brain as opposed to just recording instructions. So the smaller your devices get, the higher the spatial resolution. So it's not going to blast current into a large field. It's going to be very specific to the neurons. This is also an effort of research that's ongoing, and hopefully we'll see devices that soon allow bidirectional communication as opposed to just unidimensional communication. This is a proud moment of us. We actually have won an award for this work, but these are recordings that we did in an epileptic person. So you can see epileptic mouse. Sorry, you can see we've put 64 electrodes, 32 are on display, where you can see that every time they get a seizure, there's a lot of disturbance in the activity that's recorded. So this again gives surgeons and researchers a lot of information to study why seizures occur or why person gets epilepsy. It's also a developmental process, right? It's not that somebody just gets epilepsy overnight. So to understand what's happening, here you can see the person getting seizures. Can you imagine like the how much energy the brain is creating and why a person basically gets a seizure and starts moving erratically? So what we realized is prior to this happening, we can see some signatures. So if we can identify those signatures, we can actually stop the person from getting these seizures. For my Indian friends here, this is again, this is very interesting. But back in the day when there was folklore about ghosts and spirits entering a person's body, so this was it
Yeah.
basically. They were people with epilepsy. When they got a seizure, it kind of looked like something else was controlling them, because in this phase, there's so much energy that's in the brain. The brain is heating up that it cannot recognize or remember things. So when a person gets a seizure, they don't remember getting the seizure or anything that happened during the seizure. That's why folklore kind of suggested that, oh, there must be an external spirit that got into the person, and that's what's happening to them. And then you would get people hitting them with brooms and saying, "Get out, spirit, get out, leave this person alone." But it was actually until like the brain exhausted all its energy that the seizure went on. But this is a case of epilepsy. It's not that someone was, there's a spirit entering someone's body.
Yeah.
But anyway, this brings me to the end of my presentation. Just to show you where most of the activity is happening. A lot of it is happening in North America. There's some concentrated, particularly in Germany and the UK. And we're the only ones in India that have commercially successfully translated our devices and have been shipping overseas. And thank you for listening. And hopefully, let's do some science together in the future.
Awesome. This is great, Kaustubh. I think you really packed in a lot of technical information in a way that uh, someone non-technical like myself and maybe a lot of others on who are listening in, that was super interesting. So thank you so much for this presentation. Let's we can go in for more questions. I know people had raised their hands in the middle as well. So we are at 8:03. So also maybe 10
Yeah,
minutes more is okay. Yeah.
sounds good. Sounds good.
Okay, perfect. So more questions. So let's open it up. I think this was a lot to pack in. Me personally would love to come and see your manufacturing facilities at IIT, and maybe we can do a visit for CMU alums as well. But that's maybe sometime in the future. So,
Absolutely.
Anmol, you want to kind of like kick off the questions?
Yeah, I think we had 2 or 3 hands up. So there's Vivek and Webber. So if you guys want to ask your questions first.
Yeah, I'll go. Also, other than the technical part that was discussed, more on the business angle, I noticed that you were mentioning Neuro as a company rather than a research lab. So how did this go about on the fundraising and all that landscape? Is this more of a self-funded or a grant-based company, or this took a VC route? I mean, I'm assuming the appetite for investing in this from the VC landscape in India might be very, very low for such a sci-fi kind of product. So how did you work this part out?
That's a good question, Vivek. To be honest, it took an organic route. We didn't raise a single rupee for doing this. I was already seasoned in this business, so I had a network, and that was really my leverage. I was able to build technology, and I had a network already. So when I came back to India, it wasn't with the objective of starting a business that could grow and I could get investment and grow it further. Yeah, I just happened to be the person who everybody would come to and say, hey Kaustubh, can you make this device for me, or can you help me with these experiments? And I would say, of course I can, and I would ship out products. So we've done like in the last 3 years we've done over 1.5 crore in revenue, sorry, in profits. We've been just basically we've been running it as a business. So at this point it is an investable. I wouldn't even seek investment from others, because it's not one of those businesses where you can just grow with more money coming in. But having said that, the moment this gets translated to humans, that's where the fun begins, because at that point the whole world is your market, right? And the patient population that's waiting to get these devices is tremendous. That's why companies like Neuralink and Synchron and Paradromics are seeing the valuations they are seeing. But the key element is that you have to do work on humans. For the animal science research communities, it's a small market. It's a profitable market, but it's not an investable market, I would say.
Oh,
Vaibhav, you had your hand raised earlier. Do you want to ask the next question?
Yeah, actually, sorry. So how hard is it to get this approved for humans? Like, I've often heard conversations that people say, that you're playing God or something. So typically, how does it work? Because it seems like it could be controversial as well.
Yeah, it's a good question. So typically, when you for a device where you already have an existing treatment option that's cleared available, the route is much easier. So let's say if I want to come up with another alternative to paracetamol, I can just take a
Mm,
route and say, okay, this I have a predicate device in mind, and I'm replicating exactly what they're doing. But in these cases, there is no predicate. So whoever is the
mm,
first to do this has to go through a long cycle, basically, to get the validation for it. Some critical elements are you need to have a device that's completely biocompatible, which possesses no
okay.
risk to the patient, because the FDA in the US would not allow for it. Secondly, because it doesn't have a predicate, you have to do extensive clinical studies. So it'll be preclinical studies as well as clinical studies. And they would be done in a statistical way in a GLP certified lab.
Mm,
And at the end of this, once you have to present a case to the FDA saying, this is my technology, these are my results, these are my findings, and this is the indication of use. So while all of these technologies are platform technologies, meaning you could use the same technology base and use a different target application, the FDA requires you to get these devices approved for each individual disease that you're targeting. So if somebody is making this for Parkinson's, they would have to do all of these studies on Parkinson's patients, and the device would be approved only for Parkinson's. But in the
mm,
case of Neuralink or in the case of Synchron, they're trying to do something beyond that. So they're trying to make PCIs, and this again is kind of tricky territory. I cannot speak much on it, but it's gonna be a different model, as compared to the previous
mm,
neurological devices that were passed for very specific indications. So I think what you're gonna see is a lot of these companies are gonna pick different disease models, put their technologies, do the clinical studies, and in a matter of
mm,
maybe 5 to 7 years, you should see some feedback from the FDA whether these devices get approved or not. But until then, the jury's still out.
mm. Got it. Got it. Thank you.
Thanks. I
So,
think AK had a question.
Hi, sorry, this is Animesh. I had two
Hi,
questions.
hi, Animesh.
So what would be the, I mean, just to get some sense, how expensive are these?
Oh,
Are
I'm
they
sorry.
even
I couldn't hear
affordable?
your question. I couldn't
How
hear you very
expensive
clearly.
are these? How expensive are these today?
Oh, how expensive are these? Okay, so ooh, I have to reveal my secrets. Okay, so some of the devices I sell them, they typically vary from about $800 to $2,000, depending on how many electrodes you're packing per device. So if I go back to, a picture of my wafer, uh, it really depends on how much real estate your device is capturing on the wafer. If it's more,
So
they
you're
get
selling
more expensive.
unfinished product, right?
I'm selling what, sorry?
Animesh, I think your voice wasn't fully clear. What was the question?
Yeah, I have an issue with the network. Sorry, go ahead.
No, what was the question? He couldn't hear the question.
Yeah.
I believe that I think what
I
you
think
said
there's a
that
network. There's a
Yeah,
network
so
issue, Animesh.
Animesh, you wanna type it in?
in
I think he's dropped off.
I
Any
see.
more questions from others? Anmol, do you have any? And I think Suman Saraf also.
Hi. Yes, I had a couple of questions. So from my preliminary understanding, would it be right to say that one electrode is probably measuring about signals from about 1,000 synapses because it's like nanometer to micrometer kind of conversion? Is that
Right. Right. Well, it's hard to talk about the synapses, but we record from anywhere from 64 to 1,000 channels today. That's what is the practical feasibility, including the backend instrumentation that's used. So
right?
synapses actually vary, right? Depending
Yes.
on.
So if the synapse width is about in the order of nanometers and the electrode width is about order of micrometers, I thought from the slides. So basically, one electrode is probably capturing potential from many aggregated synapses. Would that be accurate?
No. So the distance between two neurons, which is the smallest entity in the brain, is about 80 microns. It's not nanometers. So when
Okay.
you so with these invasive techniques, you can actually record from a single neuron. That's the kind of spatial resolution you can get. But having said that, it's a good question that you touch upon. So if I give you an example, let's say Asha is recording this conversation, right? And if we give him the challenge to pick out who's speaking at what time, and everybody's speaking, let's say, at the same time, he's probably going to hear a bunch of different voices, but he will be able to kind of figure out, okay, isolate that, okay, this is the pitch of the voice, or this is the frequency of the voice, and this is where it's coming from, and he can pick out, who the person is in the room who's speaking. So it's kind of similar with electrodes as well. What you're saying is very true, that neural activity is picked up on multiple electrodes, but depending on the phase change of that signal, you can then spatially isolate your particular neuron. So that's called spike sorting in our parlance.
Understood. And these voltage or these potential changes are probably of the order of nanovolts. Is that correct? As in, you are basically picking up the potential changes or something else?
Yeah, potential changes. So they basically, there's a movement of ions, sodium, potassium, and fluorine, that create a dipole moment. So the potential that's recorded on the electrodes is somewhere in microvolts to millivolts range, depending on the interference, positive or destructive interference that they see. But it's in the microvolts range. So all of the
Okay.
instrumentation that's built is built for that range. It is a theoretical problem. In theory, we can record very clearly from these neurons.
Okay, and I had one more question. How
Yeah.
transferable are these decoder models from person to person? So, for example, a left-handed amputee and a right-handed amputee, would you detect very similar signals in, let's say, the motor cortex?
That's a very good question. So the currently from my understanding, the spike sorters are based on the designs. So if you see this picture here, this is an example of a tetrode where the distance between the electrodes is measured and it's definite. So the models that are built around this particular design will allow you to input basically the inter-electrode distance and the area of your electrode, and they can be used to extrapolate information or spatially isolate spikes. But if you change the design, then you need a different algorithm. So as long as the design stays constant, which happens in most of the cases, most people prefer a linear design, or they would prefer an edge design, or a tetrode like this, honeycomb design. The algorithms are transferable provided the design stays similar.
Yeah, but wouldn't it assume that, let's say we implanted it in exactly same regions of the brain of multiple people, the electrical activity in everybody's brains would have to be similar for the same
That's
design?
true. Yes. Yes, absolutely. They would be very similar. That's why this technology is transferable from person to person.
Okay.
So if I give you an example of someone with epilepsy, you notice that when they get seizures, there's also a behavioral aspect to it. So some people would bite their tongue, or some people would turn to their left, or some people would have their necks locked. So if somebody is exhibiting similar behavioral patterns in different parts of the world, it is very likely that the same similar areas in their brain are having challenges. So that's why a lot of neurosurgeons, so far, when treating epilepsy, neurosurgeons keep a record of the behavioral aspects. So they say, okay, this person exhibited this behavior, turned to the left, or hands left hand lock. And that's how they share notes that, okay, I recorded this at, so and so coordinates. So it is very likely that the next patient who exhibits some similar symptoms would also have them at so and so coordinates.
Great.
Yeah, got it. Thank you.
Yes.
Awesome. We're at 8:15. Anmol, do you have a question, or should we wrap up?
I can go with the last question. I know we are over time. Uh, but so you have worked in US, at two different companies, plus you've researched about markets in Australia and Europe as well. So what did you feel were the challenges and the benefits of, having built your startup in India in this sector?
The challenges, actually, it's been, contrary to what you would think, it was actually much easier for me to build things here because there is a concept of industrial estates in India, which is not so true in the West. In the West, when you want components or parts or materials, you would have to find somebody online, order them online. Over here, you just walk to the next unit and you're able to find them because the density of activity is so high. So that was a boon in disguise. And the second thing is the government is encouraging this because they realize that semiconductor is a very good way to get money to get to do exports basically, not just commercial activity within Norway. So because of that, they when I started, I registered for this program called INUP, Indian Nano Users Program, where they said we will completely subsidize all the developmental costs for your product. And once you start selling, then you start paying us money for using the national facilities. So all in all, I found it way easier to do this in India than I did in the US. But because everybody wants to make money. In the US, it was subsidized to a large extent, but not so much that it was conducive for a person like me to come in and start a company. In India, it is way easier. So you wouldn't believe me, Anmol. It took me less than 2 months to come in, build my product, and start shipping. Like by the fifth month, my products were used in the US and places like University of Pennsylvania, University of Florida, and University of Michigan.
This is great to hear,
Amazing.
and yeah, very promising for future entrepreneurs for sure.
Yeah, yeah. So sometimes it works out. It's I always tell people this that not all businesses are investable. people make that mistake where they come in thinking I need to raise money first, and only then do I start working on my product. Uh, Most businesses are not investable, but uh, it doesn't mean that you shouldn't do that business. It still gets you money, right? It still pays well. So I would say, that is another approach that kind of helped in India. I use that very like, shopkeeper approach where I'm gonna record how much I'm spending in the morning and then how much I'm making in the evening. That approach helped out quite a bit.
That's great. And
Yeah,
I think this is very promising for India and Make in India. The stories we hear are contrary, but good to serve first an experience of someone like you who's built it. So thanks so much for your time. Kaustubh, my final question, and we can close right
yeah,
then, is that what
right.
what was your experience working with Elon? What did he like teach you? And then he's in the news
He,
everywhere today,
yeah,
but
right.
I'm sure you've seen the other sides to it.
Right. I like to think of it as that part of my life where I was paying my dues. I think you can only kind of get experience or get good at something once you're pushed through the grind. Like you have to get your hands dirty. You have to stay back till like 12 o'clock in the night and find solutions to problems. And that pressure, right, that comes from working in a company that's got so much media attention that it's stressful at times, but that stress also helps you get better. So in like the first 3 months, I hated working there because I went in thinking I'm gonna lose my job at any moment. One mistake, and then the other 60 people are just waiting for you. They don't have anything to do because you messed up your part. So, and believe me, that doesn't get rewarded. It gets punished on the contrary. So it puts you to that grind, and after 6 months, when you know you kind of get in that approach gets inculcated in the mind. You say, all right, I'm going to do this, and I can say that I'm going to deliver by so and so date, and I'm going to be able to do it. So it's rough. You have to put in a lot of hours. Definitely not for married people or people who have kids, because I don't think the spouses or the kids are gonna like you spending most of your time at the company. But
Yeah.
as a bachelor, which I was when I was working there, it was fun. There were a lot of stories. There's a lot of pressure, but it makes up for it in the end. One of the best experiences I had was, I almost like went. I used to sleep like 4 hours a day during the week of the media presentation. Everybody was,
Oh,
so busy. But the day of the media presentation, Elon took us to his house, and going to sorry, not a millionaire, a billionaire's house is a completely different thing. Like you haven't seen
yeah.
such opulence anywhere else before. So that it made it worth it. So I think working with Elon was stressful. You get celebrated for doing good things, but you get punished for doing bad things as well.
Awesome. I think you've clearly benefited from it, and as has our CMU community for learning from your perspectives and seeing such great hardware manufacturing happening in the country. So thanks so much, Kaustubh.
My
I think
pleasure.
we could have gone on for another hour as well, but we will give you time with the family. We won't be Elon. We'll give you time with the family
Yeah.
and
Thanks.
and
Thanks, Asha.
kids.
If I can get one sentence in, I'd just like to say
Yeah.
that much before Neuralink, I think CMU inculcated this in me. I'm sure a lot of us, when we went to CMU, we went there as pretty lethargic, buffoons, and then the day we graduated out of CMU, we were primed for the world, right? We're primed for industry. So this is where it started. Really, CMU was where it
Awesome.
started. Wasn't it, Neuralink?
Awesome. Great to hear. I think it's very encouraging for all our alums and everyone who's joined in. So thanks again, Kaustubh, and appreciate it. Hope to meet you in person and visit your facility as well. Thank you.
Absolutely, absolutely. Thanks, Asha.
Bye
Thank
everyone. Thanks everyone for joining.
you.
Thank
Bye-bye.
you.